Here I am before you, all the way from Germany, with the great honor of introducing Iva, who is reaching a significant milestone in her academic journey today. Iva brings together the realms of microbiology and computer science through her skills and training in both disciplines. So much so that when she joined our group almost exactly 4 years ago, Iva was not 100% sure whether to give priority to wet lab or computational work. She started her PhD to make sense of the elusive human immune-modulating bacteria that live very close to the eukaryotic cells in the human ileum, which was the wet lab path. Later, she developed an interest in understanding the link between microbial ecology and the metabolic potential encoded in microbial genomes and metagenomes, which happened to be the computational path. This was quite a difficult topic and required her to code from scratch a sorely missing computational framework in our bioinformatics toolkit that can deal with complex data and models across scales, which you will hear more about very soon during her presentation. Thanks to her dedication to open science best practices, her software development efforts and meticulous documentation have already helped many researchers inside and outside of our group, even before she was able to publish a paper that described her findings. Throughout her PhD, Iva's intellect and generosity further shine through her commitment to invest into teaching efforts in bioinformatics workshops and selflessly helping researchers navigate through computational challenges everywhere. From computer code to ideas to solutions, Iva constantly shared everything openly and passionately, for which I thank her on behalf of our group and the broader community of microbial ecologists out there. I met Iva the first time at a conference while she was still an undergraduate student at the University of Illinois at Chicago. She approached me after my talk, told me that she found what she heard very interesting, and that she was wondering whether our group was looking for PhD students. I told her that if she could get into the graduate program in biophysical sciences at the University of Chicago, I'd be very happy to meet her again. Almost exactly a year after that conversation, Iva appeared in my office and said, well, I joined the program, now what? Not only Iva managed to get into the competitive graduate program in biophysical sciences, she also did it as an awardee of a prestigious National Science Foundation Graduate Research Fellowship, which means among all the institutions and groups throughout the US that would have received her with open arms, Iva decided to join our group. This will remain as one of the biggest achievements of my career in science. Looking back, I'm extremely proud of what Iva has been able to accomplish throughout her journey with us so far, and I look forward to her future endeavors and her talk. Iva, the stage is yours.
Thank you so much, Meren. Hello, everyone. Thank you for coming so much. And today I'm going to be defending my thesis, which is called A Novel Framework for Metabolism Reconstruction, Identifies Determinants of Microbial Resilience in the Human Gut. And I would really appreciate it if you could all hold your questions until the end, just for the sake of time. So thank you for that. All right, I'm going to start by telling you a bit about the open questions in the gut microbiome field that my work has tried to address. Then we're going to get into the concept of metabolic independence, why it's a determinant of microbial resilience, and how to estimate it. I'm going to spend most of my talk describing the main project of my PhD, which is the application of the metabolic independence hypothesis to inflammatory bowel disease. And at the end, I'll do my best to impart my perspective on the future of this field. So without further ado, why do we care about the gut microbiome? I don't think it's going to be a surprise to anyone here that the gut microbiome plays a role in human health, which is mostly mediated by the metabolic activity of these microbes. Gut microbes synthesize some essential amino acids. They provide us with vitamins like biotin and cobalamin. They make immunomodulatory molecules and compounds that regulate host nutrient uptake and appetite. And some gut populations produce short-chain fatty acids, such as acetate and butyrate, which are important energy sources for intestinal epithelial cells, and which also play a role in regulating gut barrier function and immune system responses. Under normal circumstances, they also outcompete opportunistic pathogens, keeping you from getting sick. And beyond all of these beneficial functions of the gut microbiota, there are also a lot of human diseases and disorders in which the gut community is altered, which has led to a lot of studies investigating whether or not these microbes have a causative role in disease pathogenesis. In light of that, there's also been lots of hype about manipulating the gut microbiome to influence our own health, such as via probiotics or therapies like fecal microbiota transplant. Our focus on the gut microbiome is reflected in how much funding the NIH is throwing at it. So out of all the body sites with microbiomes, the gut gets the most public funding, and most of that money goes towards studying its relationship to disease. I think these numbers in particular demonstrate nicely our relative lack of attention to the microbial side of the story in this field. We naturally think a lot about how our gut microbes affect us, how they support our health or give us disease, but we think less about how our health status and our actions affect our gut microbes. From a microbial perspective, the gastrointestinal tract is just the environment they live in, and that environment can support or stress out microbial populations. We can consider this as a question of microbial ecology. What are the ecological principles governing gut microbial communities? What causes them to thrive, and what causes them to decline? You probably know some of the answers to this already. For instance, we know that when you take antibiotics, a lot of the microbial community in the gut will be wiped out. Here's data from a study on the species richness of gut communities over time following a 4-day course of antibiotics. You can see that the diversity drops, and in some cases can take as long as 6 months to recover. We also know that gut microbes feed each other. For instance, when one microbe degrades complex carbohydrates into formate, the formate is then taken up by another population that can convert it into acetate. And bifidobacteria species can degrade starch into lactate, which can then be converted into butyrate by other microbes. This is called cross-feeding, and it's an important ecological support network between microbes in the gut. We know that it's important because when people get sick and the community structure in their gut is altered, cross-feeding relationships start to disappear. And this was demonstrated in a recent preprint by Marcelino et al. The bar graphs here on the side show all the metabolic compounds that have significantly less cross-feeding interaction during disease. For instance, hydrogen sulfide, which has been previously associated with symptoms of inflammatory bowel disease, had the highest loss of cross-feeding interactions in their IBD sample group. You might notice that this could imply a causal relationship between disease and loss of cross-feeding interactions. But before we get ahead of ourselves, I just want to emphasize that it's very hard to obtain direct evidence of cause and effect in most studies of the human gut, since there's such a close association between the human host and the microbiome. Inflammatory bowel disease provides a good example of this. In IBD, microbial diversity decreases in the gut. Does the diversity decline first, and that shift causes IBD? Or is this decline a reaction to the other causes and downstream symptoms of IBD? Most likely, there's some sort of positive feedback loop between the disease manifestation and the biodiversity decline, which combine to together contribute to a decline in host health. But we still don't really know what comes first, and more importantly, we don't know whether we can do anything with the microbiome, now that we have the ability to modify it, that will ameliorate disease. So all we can do in these situations where you have diseases of complex etiology so closely intertwined with the fate of the microbiota is to suggest a model for how we think it works and see whether the data supports that model, which is what people have been doing so far with IBD. And it's led to this paradigm in which microbial dysbiosis in the gut is essentially considered to be at least partially responsible for a variety of human ailments, from IBD to obesity. But like I said before, there's been a lot of attention to the human side of the story so far. What's happening to the microbes in this picture? And that's where my PhD project fits in. I've been investigating the role of microbial metabolism in determining biodiversity of gastrointestinal communities during disease. Or, put another way, what metabolic capacity do microbes need in order to survive in the face of disease-related alterations to their environment, such as inflammation? And beyond disease, I hope to convince you that these metabolic determinants may actually apply more generally as determinants of microbial resilience in the face of different kinds of environmental perturbations or stresses to the gut microbiome. I'll show you some evidence of that later using data from the study on antibiotics that I just showed you. But before we get into all that, I know not everyone here is in this field, so I briefly want to mention the nature of the data. Like most studies of the gut microbiome, I used human fecal metagenomes in which you sequence all the DNA that you can find in a poop sample. Now, poop is a convenient medium since it's rather easy to collect, but keep in mind that it can only tell you about the colonic microbiota. With the DNA in a metagenome, I could assemble pieces of different microbial genomes, and from there, I could identify genes encoding enzymes and match those enzymes up to metabolic pathways. And I'll give you more details on this part later. But first, I want to provide an answer to our ecology question, the metabolic independence hypothesis. And this is actually coming from work done by Andrea Watson during her PhD. Some of you might even remember her thesis defense a few years ago. And that work was also very recently published in a paper that was co-authored by Andrea, Jessica Fussell, and myself. In this study, we investigated colonization in the gut using fecal microbiota transplant, or FMT, as a model system. For anyone who doesn't know, FMT is when stool from a healthy person is transferred to the GI tract of an unhealthy person as a way to sort of reset the gut microbiome. So what Andrea did is to analyze a time series of fecal metagenomes from stool donors and stool recipients before and after FMT, and then she used genome-resolved metagenomics to track which microbes from the donor's healthy gut could colonize the recipient's unhealthy guts. So some microbes were able to colonize, but others did not. And when we looked into the functional differences between these two groups, we saw that the colonizing microbes had more metabolic capabilities than the ones that didn't colonize. So their genomes contained a lot of complete metabolic pathways, especially for biosynthesis of amino acids, cofactors, vitamins, nucleotides, while the non-colonizing genomes did not encode these pathways. So in other words, the good colonizers could make a lot of stuff for themselves, and we therefore labeled them as having high metabolic independence, or HMI, while the poor colonizers had low metabolic independence, or LMI, since these guys would have to rely on uptake of critical metabolites produced by the other members of their community, rather than making things for themselves. We figured that if metabolic independence can determine colonization success when microbes are being transferred from one gut to another, perhaps it can also determine microbial survival in the face of environmental stress, such as inflammation during IBD. So we started looking at genomes reconstructed from healthy gut metagenomes and from IBD gut metagenomes to see if we were right. It turned out that the healthy guts had a mix of HMI populations and LMI populations, while the IBD guts had mostly HMI populations, which would seem to support this idea that metabolic independence might be critical to microbial resilience in IBD, since only the HMI microbes seem to survive in the inflamed gut environment. And this is the actual data that demonstrates this. On the top here are box plots of pathway completeness scores in different samples, and on the bottom are histograms of the same data, each for a single pathway. You can see that the metabolic completeness scores are generally much higher in the genomes reconstructed from people with IBD, which are in red and orange, than in the genomes that were reconstructed from healthy people, which are in green. So this study was a proof of principle for us that high metabolic independence is a determinant of biodiversity in IBD, but it had a few limitations. So first, since we needed very deep sequencing in order to be able to reconstruct genomes, we had a fairly small sample size. And furthermore, very few studies are reporting genomes reconstructed from IBD samples, so we didn't have a lot of genomes from different sources to compare. We wanted to scale up this analysis by comparing metabolic capacity encoded in a large set of publicly available gut metagenomes from healthy people with those from people with IBD. However, there's one more problem. We did the previous analysis by reconstructing high-quality genomes from fecal metagenomes, and then we estimated metabolism on a genome level. But metagenomic binning is difficult, and it's very time-consuming if you want to do it well. So scaling up our analysis to hundreds of metagenomes is really only tractable if we avoid the binning step and instead look at community-level metabolism, using that data to make inferences about individual microbial populations within the community. In order to do that, we need to be able to estimate metabolism directly from metagenomes, and that estimation has to include a metric for pathway redundancy, that is, copy number. Why copy number? Because metagenomes contain lots of microbial populations in them, and all of their collective genomic content will be used in the estimation process, you can't rely on pathway completeness scores because those will all be overestimated up to a maximum of 100% complete. So we need to estimate pathway copy number instead, because those values will scale proportionally with the number of microbes represented in the metagenome. Well, doing this required some technical advances because although there are lots of existing software tools for metabolism reconstruction, most of these only work on genomes. Out of all of these tools, only 2 of them work on metagenomes, and only 1, pathway tools, has a redundancy metric for pathways. And in the case of pathway tools, the redundancy metric is pathway abundances, which also may not scale linearly with number of microbes in the metagenome because pathways from more abundant microbial populations will have higher abundance. Or in other words, there is an unequal contribution from different populations to the abundance of the pathway as a whole. As a result, a large part of my PhD work has been developing a software framework for metabolism reconstruction, which works on both genomes and metagenomes, and which can calculate pathway redundancy in the form of copy numbers, in addition to the standard completeness scores that I was talking about before. I won't bore you with all the technical details of this software, but I do have diagrams to explain it. So if you're curious about how the computation works, just ask and we can come back to this slide to discuss it. But I do want to mention one more technical detail before I go on with the science, and that is normalization. In my work, I'm comparing the healthy samples with high diversity to IBD samples with low diversity. Since there are so many more populations in the high diversity samples, pathway copy numbers will all be higher in those samples, while in the low diversity samples, the estimates will naturally be lower. For instance, you can see that in this scatter plot, that most pathways have higher copy numbers in the healthy sample group. So here the x-axis is showing median copy number in the IBD samples, while the y-axis is showing a median copy number in the healthy samples. Most points are above the line, which is as expected because there are more microbes in these samples, contributing to higher counts of each pathway at the community level. But as I said, in our case, we care about what pathways are important in the IBD samples for individual microbial populations. So in order to answer our question and make these copy numbers comparable across samples with disparate levels of biodiversity, I needed to normalize pathway copy numbers. I do this by dividing the copy number value of a given pathway with the number of estimated microbial populations in a given metagenome sample. To get this denominator, to estimate the number of microbes in a given sample, I count the single copy core genes. These are genes that should only be present in one copy in every single microbial genome. And I take the mode, or the most common value of those counts, and use that value to normalize the copy numbers. After normalization, I will end up with a per population copy number, or PPCN, which is a better metric for comparing metabolic capacities given the differences in biodiversity between samples. And going forward, when I talk about copy number, I'm mostly going to be referring to per population copy number. So now I think we're ready to talk about my main project, investigating the relevance of the metabolic independence hypothesis to the gut microbiome in people with inflammatory bowel disease. As I mentioned, I wanted to generalize the previous analysis that I did in our FMT study by scaling up to a large meta-analysis of publicly available fecal metagenomes, which were all published in the studies shown here. These studies cover a variety of human health conditions, but for most of them, I'm using only samples from the healthy controls, representing healthy people. And for all of the studies that are concerning IBD, we have samples from people with IBD, as well as samples from people who are not clinically diagnosed with IBD, but who are also not necessarily healthy. And that pink group of samples is called non-IBD. For the most part, I'll be focusing on the differences between the healthy group and the IBD group, but the non-IBD samples will come in later. And just a quick shout out to Emily, who downloaded and assembled many of these metagenomes. She used them also in her own project, which she's defending on Thursday, and you should all go see that. I'm sure she'd love to see you all there. Collectively, all of these studies give us about 2,800 samples. But unfortunately, a lot of those samples have very low sequencing depth. This is a problem because to accurately estimate the number of microbial populations in a sample for estimating the number of populations and normalizing our data, we need a high enough sequencing depth to assemble and capture all of the populations, especially those from the rare biosphere. You can see here that as sampling, the sequencing depth increases, the estimated number of populations is also increasing. But at some point, it starts to plateau once there's enough depth to capture almost all the microbes. So I decided to set a sequencing depth threshold at that point, keeping only samples with at least 25 million sequencing reads so that we can normalize accurately. And that leaves us with about 400 samples to work with. The sequencing depth thresholding makes a few of the studies drop out of the analysis, and those are shown in orange here, but all of the other gray studies in this table were used. And you can see that they cover a wide variety of cohorts from different places with different health conditions, ages, and genders. And that's important to note because the results I'm going to show you are describing a general, high-level trend that's holding true across a diversity of individuals. And I just want to emphasize again that proper normalization is critical here. I'm going to show you how the unnormalized data compares to the normalized data so that you can see the difference for yourself. First, on the slide are several plots of the unnormalized copy number values. On the left is the scatter plot I showed you earlier, which shows that most pathways are overall increased in the healthy group. And each point here is a median copy number for one pathway aggregating over all of the samples in a given group, IBD or healthy. But in the middle is a heat map where you can see all of the individual copy numbers for each pathway in each sample. The pathways are in the rows and the samples are in the columns. In darker colors, since it's a heat map, indicate higher copy number. However, there isn't really a clear signal differentiating between the healthy and IBD sample groups, even for those pathways that we know from the FMT study to be associated with high metabolic independence. Those HMI pathways are in these box plots on the right, which shows the distribution of their copy numbers in each sample group. Overall, the only observation we get out of the unnormalized data here is that these pathways have higher copy number in the healthy group, as we would already expect, given their increased diversity in those samples. But now let's look at the normalized data. So here on the bottom are the equivalent plots using per population copy number instead. And now a signal becomes clear. So most pathways are in fact having slightly higher per population copy number in the IBD metagenomes. And for a certain subset of metabolic pathways, the ones in the box down here in this heat map, per population copy number is significantly higher in the IBD sample group. These include those pathways for high metabolic independence. So the normalization is critical because without it, the difference in sample biodiversity drowns out this relative population level metabolic signal. To figure out which pathways were the most important to IBD gut microbiome, that is the ones in this box right here, which are most increased in IBD compared to the healthy samples, I conducted an enrichment test on the normalized copy numbers in each group. After thresholding for both p-value and effect size, here's the final list of IBD-enriched pathways. There are 33 of them, and on the right, you can see a box plot of their median normalized copy numbers grouped so that you can see their differential distribution between healthy and IBD. And if you look at the identities of these pathways, you might notice that 70% of them are for biosynthesis. Now, thinking back to the FMT study data that I showed you earlier, a large number of the important pathways there were for biosynthesis as well. So this result supports our hypothesis that microbes with high metabolic independence, i.e., those microbes that can produce a lot of critical metabolites for themselves instead of relying on those molecules to be shared by their community, these HMI microbes are more prevalent in the IBD gut microbiome, thereby suggesting that they have a survival advantage in this environment. I then looked at the non-IBD samples to see where they fit in. Now, these samples are from people without a clinical IBD diagnosis, but most of them are not healthy. They either have subclinical symptoms or colorectal cancer. And their copy numbers for those 33 IBD-enriched pathways are intermediate to the other groups. This is interesting because it suggests that the proportion of microbes with high metabolic independence is increasing along the health gradient from most healthy to least healthy. To confirm this, I organized all of the samples in each cohort on a gradient from most healthy on the left to least healthy on the right, using the cohort descriptions and exclusionary conditions listed in each study. And here I'm showing you median normalized copy number data from each sample in this order. There are a few exceptions from the cohorts with very few samples, but in general, you can see there's a trend that copy number increases as host health worsens. So it seems that high metabolic independence in the gut microbiome goes beyond simply differentiating between people with IBD and those who are healthy and can actually indicate the degree of host health. This will be important later, but let me pause for just a moment to summarize the results so far. We have 33 metabolic pathways that are enriched in IBD that are mostly biosynthesis pathways and therefore are associated with high metabolic independence and that increase in copy number as host health worsens. These results are emerging from the metagenome level metabolism estimation data, which is coming from the novel methodology for calculating copy numbers and for normalizing those copy numbers that I developed. So I wanted to confirm if we get the same results when we look at individual genomes using more standard methods. I did mention before that not many studies are publishing genomes reconstructed from the human gut environment, but what we do have access to is a large number of good quality reference genomes from the Genome Taxonomy database. We just need to figure out which of those are associated with the human gut and relevant to our question. To do that, I leveraged Matt Schechter's EcoPhyla workflow to identify genomes that could be from gut microbes based on the detection of their single copy core genes in healthy human gut metagenomes from the Human Microbiome Project. And then I narrowed down that set further to those genomes that I could sufficiently detect in my set of healthy and IBD metagenomes. In the end, I got 338 GTDV genomes, which represent gut microbes, and I estimated metabolism for those genomes. This time, since these are now individual populations rather than whole communities, I estimated metabolic completeness scores rather than copy number. I specifically looked at those pathways that were enriched in the IBD sample group, the 33 pathways that are associated with high metabolic independence. And I used their completeness scores to identify which genomes are metabolically dependent or not. I basically added up all of the completeness scores for these 33 pathways to give each genome a single score, and any genome with a score that was high enough to indicate each pathway was, on average, at least 80% complete, was considered to have high metabolic independence. That gave me two groups of gut microbes to compare, which I'll refer to as HMI genomes, which are highlighted here in red, and non-HMI genomes, which are not highlighted. To see if I could recapitulate the metagenomic results with these genomes, I compared the distribution of each group across the healthy and IBD samples using read recruitment data from the metagenomic short reads to all of the GTDV reference genomes. When I looked at the detection of each genome across samples, I saw that the HMI genomes have significantly higher detection in IBD samples than non-HMI genomes do, and that's here in the red box plots. But the two types of genomes were similarly distributed in the healthy metagenomes in blue. And this is what we expect to see under the metabolic independence hypothesis, since it suggests that there is some sort of selection for metabolically independent genomes in the IBD gut environment, but not in the healthy environment. Furthermore, when I looked at each sample independently, and I counted the proportion of HMI genomes detected in each individual fecal metagenome, the IBD samples had the highest proportion, the healthy samples had the least, and the non-IBD samples fell in the middle. So again, our results are demonstrating that high metabolic independence is increasing proportionally with host unhealthiness. So our analysis of reference genomes has confirmed our metagenome level results. The microbial populations with HMI are enriched in the IBD gut environment. But can we use this information for a practical purpose? For instance, to diagnose IBD and related conditions from fecal samples. As it turns out, you can. So when I trained a logistic regression classifier on the copy number data for those 33 HMI pathways, it performed quite well. The blue line in this plot on the left shows its average performance in a 25-fold cross-validation test compared to this pink dotted line, which shows the baseline performance of a random guess. In these sorts of plots, the closer you are to the top left corner, the better your model is. So ours is pretty good. And the heat map on the right shows you the proportion of correctly and incorrectly classified samples in each fold of the cross-validation. About 90% of healthy samples and 77% of IBD samples were classified correctly, for an overall accuracy of 86%. But let's not just stop there. I want to remind you that we're looking at an ecological phenomenon here, in which gut microbiomes, under the stress of coping with their host's IBD symptoms, are filtered such that the surviving populations are mostly those with high metabolic independence. This filtering gets stronger as the environmental pressures get stronger, as shown by the increase in HMI across the gradient of host health. And it doesn't appear to be specific to inflammatory bowel disease, considering that we also see this gradient with our non-IBD samples, which are mostly coming from colorectal cancer patients. So perhaps we can generalize this phenomenon to other forms of stress on gut microbes. In other words, rather than differentiating between healthy people and people with IBD, perhaps we can more generally differentiate those whose gut microbiomes are under stress versus those that are in homeostasis. And I'm going to show you this using data from the antibiotic time series study that I showed you earlier in the talk. Now, this study took 12 healthy people and gave them broad spectrum antibiotics for 4 days. And they used fecal metagenomes to track the response and recovery of the gut microbiome over time. Here in the bottom is a plot of the species richness over time, which I showed you earlier. And you can see that following the antibiotic treatment, the gut microbiota declines and then eventually starts to recover. When I classified these samples using our logistic regression model, I saw a very similar pattern over time in the classifications. In the initial time point, before antibiotic treatment started, all of those samples were classified as healthy. Then, the proportion of healthy classifications decreased to a minimum 8 days later, so 4 days after the antibiotic treatment stopped. And afterwards, as the microbiomes recovered, the proportion of healthy classifications also increased. So this is a nice indication of the generalizability of the concept of high metabolic independence, not just to disease, but also to antibiotic. What I've shown you so far is that gut microbial populations with HMI are enriched in individuals with IBD, and also that the same phenomenon occurs following antibiotic treatment. If we combine this with what we already know about the loss of ecological relationships within gut communities in individuals with disease, we arrive at a new model for interpreting the loss of diversity in human disease states. When the gut community is continually disrupted by stresses such as drugs, disease symptoms, or their host's overactive immune system, the community interactions are also disrupted, and therefore only the microbes that can produce critical molecules for themselves can survive, while the microbes that are dependent on community resources do not make it. This is a neutral process of ecological filtering, which results in a microbiome that has adapted to survive the relative lack of ecosystem services typical in these conditions. Under this model, loss of microbial diversity is not necessarily a prerequisite of disease pathogenesis, but it may eventually have downstream effects that worsen host health, like via the positive feedback loop that I alluded to earlier. So these results don't necessarily contradict the existing data on the gut microbiome's association with human diseases. They simply offer a different perspective with which to interpret that data. And if you're interested, you can read the full story in our recent preprint. Before I stop for today, I just want to spend a few minutes discussing where I think the field of microbiome research is going. More and more work in microbiology is computational, and I think that presents a really great opportunity for us to increase our use of open science practices. What that means in practice is adhering to the FAIR data principles, making methods available in reproducible workflows or on protocols.io, writing open source software, and making results available to the public in preprints, all to enhance reusability, reproducibility, and accessibility in microbiome research. I've seen the benefits of open science firsthand during my work here. DNA sequencing, for instance, requires specialized equipment and personnel and costs a lot of money, in addition to the fact that you actually have to find people to donate samples to you in the first place. But all of the data I showed you today was coming from publicly available sequences. So I didn't have to spend any of my own resources or time trying to generate my own metagenomes, and that sped up my research quite a bit. It also came with a variety of unique frustrations because not all of the data that is out there is easily accessible or well described. Missing or unclear data and metadata can really detract from its utility, which is why the FAIR data principles are such an important initiative to consider when going open science. I also think that doing science openly helps to build a support network for researchers, and this is especially important for those who have fewer resources available to them. Open science manifests in open communication via tutorials or reproducible workflows that people can use to learn new computational techniques, or through messaging channels where they can ask for help from like-minded experts. I also mentioned open source software, which is really important to making computational science accessible and transparent. I developed my metabolism tool openly as part of the Anvio platform, and I updated its online documentation as I did so, which means that other people could start using this tool without my help long before I wrote a paper about it. There have already been 25 peer-reviewed studies that mention using my tool to do their science. These are just some of the examples. And I'm not involved with most of these. For those studies that I am involved with, working with external collaborators so that they could use my tool more effectively really helped me to improve the software by exposing it early to users with a variety of perspectives and needs. And speaking of which, this really wouldn't have been possible without the existence of the Anvio platform and the hard work of all of its developers who are dedicated to keeping it accessible and relevant to microbial omics research. So a big thank you to all of these. I know not everyone has the resources or support to be able to do science openly, and I'm immensely grateful that I've been able to do so because I really think it's an important future direction for the microbiome field. And it's one that may even be necessary for researchers to keep up with ever-advancing technologies. And that's all I have to say. Thank you so much to my thesis committee, my advisors, my program, my funding, my lab, and all of you for supporting me in this endeavor.